Let's take a look at miscellaneous information. So we have 5 questions here, and these will show up again at the end of this, which is pretty brief video. How do embryonic and adult stem cells differ? So we will talk about stem cells here in the beginning. What are capsids? So we'll go over capsids. Result of the Lytic cycle. Neurodegenerative diseases in mammals are called what? And describe the Dahle experiment. Okay, so let's go ahead and get started. In animals, embryonic stem cells differ from adult stem cells in that embryonic stem cells are totipotent and adult stem cells are pluripotent. Totipotent stem cells have the potential to develop into any cell found in the human body, while pluripotent cells are known to develop into cells and tissues of the 3 primary germ layers. It is true that stem cells can differentiate into specialized cells. We forgot to put a bullet point here on a quick point about cancer. Random spontaneous mutations, mutations caused by X-rays, and transpositions can contribute to the development of cancer. So you'd like to know that. Muscle cells differ from nerve cells, mainly because they express different genes. Viral genomes are surrounded by protein shells known as capsids. So I believe capsids was one of our questions, was it not? What are capsids? Sure. Okay, so we'll talk about this for a second. A capsid is the protein shell of a virus, enclosing its genetic material. It consists of several repeating structural subunits made of protein called promoters. The observable 3-dimensional morphological subunits, which may or may not correspond to individual proteins, are called capsomeres. The size and shape of the capsid dictates the size of the genome. In the Lytic cycle, a large number of phages are released at a time. The Lytic cycle results in the destruction of the infected cell in its membrane. Prions are misfolded versions of normal protein which cause TSE. TSE are neurodegenerative diseases in mammals. A part of the promoter, called the totipotent, is said to be highly conserved in evolution. Highly conserved means that it has remained essentially unchanged throughout evolution. Therefore, any mutation in the totipotent is extremely unlikely to be expressed because of its highly conserved characteristic. And then finally, with the Dolly experiment, fusion of an adult cell's nucleus with an enucleated sheep egg, followed by incubation in a surrogate, was used in the revolutionary Dahle experiment. In 1996, Dolly the sheep was the ultimate product of a series of cutting-edge cloning experiments that would soon rock the research world. Animals had been created from embryonic cells before, but Dolly was the first mammal successfully cloned from an adult cell. Her birth made Dolly and the scientists who created her instant celebrities, and the wider public realized that science fiction vision of factories turning out hundreds of clones as a way to preserve loved ones, or to provide medical spare parts, or simply as military cannon fodder was now technically feasible. Okay, and then we jump back in to our 5 questions. So this is a great time. Hit that pause button and see how you do on answering the questions for yourself.